So what I'm going to do is I'm going to take you through a demonstration that I did, basically what I did with my high school class that I've been teaching at Green Valley Ranch for the past 2 years. And this isn't some special school. 60% of the kids there get free lunch. It's 95% of the kids there are minorities. In my class, I had 7. 3 of the students didn't even get to walk down graduation because their grades weren't up to par. I'm sure some of you are going, I bet I know those kids. You'd be surprised. Actually, one of these kids was probably the smartest high school kid I've ever met, and he's also an undocumented immigrant too. So what I'm going to do is I'm going to give you, I wasn't kidding. I teach this to high school kids, and if I can teach this to these kind of kids, you guys can get this as well. So I asked him, I said, the world's research is available to us. Anything you want is out there. We've got 2 million different samples to choose from. What do you want to do? One of the students, said, hey, I had an aunt that I really liked. She died of breast cancer 2 weeks ago. I go, well, you know what? Let's look at breast cancers. Let's see what we can find. So basically, and that's what you do is if you're going to analyze a data set, I went in, basically explained what breast cancer is, some of the things behind it. And I also wanted them to understand that anything you look at, any condition, it always kills me. Like I'll analyze, somebody will say, hey, they'll do a RNA-seq study and they'll look at basically the expression of all the genes, in that genome, not only the ones we know, but some of the stuff we don't even know. And then, you analyze it for them and you come to them and they say, what's the answer, right? But this stuff there isn't an answer. Usually what I'll do is just blurt out 4 and see what they say. Usually they just go, what, really? No, it's a story. Biology is a story. And that, that's, when we're talking about breast cancers, there's 2 big forces that we're concerned with. It's the oncogenes, which are kind of the criminals in your cell, right? And the tumor suppressors, which are the ones, basically the genes that take care of the criminals, right? And we can talk about the criminals. I basically went into the class, so we're going to analyze this data set. We're going to look at breast cancer. Here are the things that kind of cause breast cancer, growth factors. What do you think the biggest growth factor to breast cancer is? Estrogen. Somebody said it. Yes, we're going to see that. Thank you very much. By the way, does anybody work on breast cancer here? Darn it, because you're going to get some free information. So I usually charge big money for that. Growth factors, signaling molecules, cell cycle genes, things that grow blood vessels, angiogenesis, things that maintain chromosomes to allow the cell to grow. And we already have some targets here, right? KRAS, EGFR, MYC, this is an oncogene, a transcription factor. VEGF, this grows blood vessels. Another important thing to realize too is that if you're analyzing a disease, right? I call these oncogenes, and I put them by the criminal, but are they bad genes? No, right? They are now, but they don't start off that way. So a lot of times in disease, what you're looking for is you're not looking for what's established. If everything was working correctly in your body, you wouldn't have the cancer, right? It's when things go wrong and things don't work kind of like, what we're expecting. And, that's what you're really looking for. And then again, we talked about tumor suppressors, cell cycle inhibitors, immune response, and that's a big one. That most of us in our room, actually all of us in our room has had cancer, right? That you've had cancer, but your immune system took care of it before it became a thing, before it grew and became a problem. It basically killed it. If you're going to have advanced cancer in your body, somehow this cancer has to hide from that immune system. So that's a big thing about these oncogenes that we're looking at. Telomere masons, again, apoptosis, when things go wrong, you need to shut the cell down. Something goes wrong with these in cancer. And here's just some things that some tumor suppressors we know. But again, what I want them to understand, and basically anybody that works in bioinformatics, is it's just not one gene. It's usually a pull of two forces, and it's not just one thing, it's lots of things. And that it's not just this one winning or this one winning, it's somewhere in the middle. So we're going to set out, we're going to look at breast cancer. You know, we started doing our research, and what we found was, you know, you can't just say, do a research project where you say, oh, I'm just going to take a bunch of breast cancer cells and look at normal tissue and see what I find. Everything is going to be too different. So bioinformatics works best when you actually ask a very specific question. So we were looking on here, you know, we were looking, we're saying, well, where can we make the most maybe impact? If you notice from stage 2 to stage 3 breast cancer, there's almost a 50% or about 40% drop in the survival rate. We thought, okay, well, let's specialize here. What's different? What happens when it goes from grade 2 to grade 3? What does it change that makes our survival rate so terrible? And also, one of the nice things, so we had our problem, and what I wanted to tell them, and I come across this all the time, is that, you know, just using this breast cancer as an example here, you know, 70 to 80% of breast cancer cases, we have no idea how it's caused. There is a lot we don't know, and it always amazes me. You know, I'll tell you right now, I do like my job, because I get to investigate all kinds of different diseases, and every time I get into a disease, it's a different thing that I learn. And a lot of times, as I get into these projects, like I was just kind of messing around with sickle cell anemia, some of the data sets, you know, and I found transcription factors that no one even thought were, you know, associated with the disease. Lots and lots of gold in here, and that when somebody publishes on their data set, say they did an RNA-seq study and they do their publication, they're only using, you know, think of it as an iceberg. They're only using this amount of the information on that publication. You've got all of this that you can still use. These data sets, other people's data sets, are gold mines, because number one is you didn't have to generate yourself, and you didn't have to pay for it. Okay. So let's get to the example here. Probably like, okay, enough with the stories.